Hello everyone, welcome back to my channel Agrotech once again. You can follow me with now Facebook, blog account and Twitter for more updates. Plus, I will appreciate your feedback for my channel as well. Today I am here with methods that involve in genetic engineering. But before going to the content, if you haven't subscribed my channel yet, please do subscribe now and please do not forget to hit like and share button as well. Well, guys, I'm available on Facebook as Agrotech. This is my Facebook page. You can find me as Agrotech official group there. Please, guys, do not forget to like my Facebook page, as my Facebook page is directly linked with my YouTube channel. You can get all of my videos from that page. Plus, if you have already followed my blog account, you will be get update from my blog article as well, and you will be getting all of my YouTube content there. So guys, please do not forget to like my page. And well, guys, I am also available on blog account as Agrotech. You can find me there as agriculturaldream.blogspot.com. So please, guys, you will get all of my article there if you follow me and if you subscribe me using your email address. So guys, do not forget to follow and subscribe my blog account as well. Let's move on to the actual content, which is the methods that involve in genetic engineering. And there are some methods, and I'm going to separate them. First is cloning. This cloning includes either plasmid method or vector method. What happened in plasmid method is we make recombinant DNA using the plasmid of vector DNA. What we do is we transform the genetic material of one organism to the another organism, or we incorporated some foreign DNA, foreign genetic material into that DNA and make a recombination. So maybe you are already overwhelmed with recombinant DNA, so I'm going to quickly skim this. First, we isolate the DNA fragment of desired DNA, or we isolate the desired DNA to be incorporated. Then we make the recipient vector plasmid ready to accept this donor DNA. We introduce this donor DNA into the vector plasmid. Then we make a recombination and we let them go to replicate. Then after that, what we do in plasmid method is we have to transform these recombinant DNA into the specific host cell, bacterial cell. So I'm taking here an example of E. coli, which is a model organism, model bacterial cell for most of the genetic engineering studies. So once we have to transform our DNA into the bacterial cell, we have to let them grow in plate. And for E. coli, we have to use LB media, and once we follow this specific procedure, we will get some colonies, bacterial cell colonies. Then we have to culture these cells in a specific media. Then we have to extract cell, and then we have to extract DNA. And then finally, we can confirm if we have done our job in right way. It means once we extract DNA, we can verify in two different ways. One is using the restriction digest, or by doing the PCR, we can confirm if we have successfully made the recombination. And once we got the once we do the restriction digest or PCR, we load the DNA into the gel. I'm taking agar gel as an example here, and we compare the DNA band with marker, and we verify if we have successfully developed the recombinant DNA. In this way, we make the recombinant DNA using the plasmid method from the donor plasmid and incorporating into the recipient plasmid. But the vector method is something different. What happened here is we isolate the gene of interest from one organism or the same organism, and we directly introduce these isolated gene into the either virus or the bacterial vector cell. Here in this vector method, in a similar way, what we do, we isolate the gene, then we prepare the vector cell, either bacterial or viral cell. Most commonly virus is using for the vector method. Viral cells are most common for the vector method, but sometimes we use the bacterial cell as well, and we introduce them and we let it to be recombine. The vector cell, either bacterial or viral cell, after introducing the isolated gene of or isolated DNA of interest, will let it grow inside that cell, and it will carry the different genetic material into that cell. And after the recombination, we let it grow, we culture in like in similar way as we did in bacteria, and we extract cell and we extract DNA, and finally we conform. And in a similar way like we did in the plasmid method, but the virus, if we use the virus as a vector cell, then we should have to like follow the specific procedure and the specific media to grow. However, like it's a similar, but in this method, the bacteria or the virus, it act as a carrier for a gene or genome carrier for the different organism or the same organism. And here we have the another method, which is called Biolistic method or gene gun method, specifically designed for plants. What happened in this method is a gun is used to penetrate a plant to introduce DNA from different organism or different plants or different source to make it genetically modified. What happened here is, the DNA stick into the specific metal surface, such as gold or tungsten. And once we stick DNA into the metal plate, we can place these metal plates into the gun and we fire into the target section into the plant, and we can make that plant genetically modified. This is a gene gun method, and this is highly successful or being more practiced in plant, but scientists are using or exercising these method into the animal cell as well. And final one I would like to call as modern methods, which deals with mutation. And there are specific some examples or some tools they are being successfully used for gene edition. First one is CRISPR-Cas9, second one is TALENs, and third one is ZFNs. And CRISPR-Cas9 came as a blessing for most of the scientists, with like being able to find most of the treatments for cancers. And scientists are working for HIV to develop vaccine as well using the CRISPR-Cas9. What happened in CRISPR-Cas9 is the Cas9 is an specific enzyme. Is a specific enzyme that in corporation or in integration with RNA guide form a specific component, and it got stick to the DNA fragment. And once it got stick into the DNA fragment, it identified specific DNA sequence. So once it identified that DNA sequence, we can delete the both strand of DNA there. And once we can become successful to delete that sequence, we either can incorporate other DNA from different source, or we still can remove them. In this way, CRISPR-Cas9 work in edition. And another ones are TALENs and ZFNs, I mean zinc finger nucleases. They do have also specific type 2 restriction enzyme, which is called FOKL. Then this FOKL act in different way for TALENs and ZFNs, and the only like operation they do is they cut the DNA fragment from both strand and make the sticky ends. So in these sticky ends, we can incorporate other DNA, and we can introduce other foreign DNA there, and we can make a recombination, or we simply can like cut and edit. In this way, these three methods are successfully using in gene editing, and these are like becoming more successful on edition tool later on. in the cancer cell and some other treatment. So these are the common methods of genetic engineering being used in this era. This much for today, and if you want to know more about the what is PCR, cloning, gene like plasmid or genomic DNA extraction, markers, anything in the future video, please get tuned with my Agrotech channel, and do not forget to like and share and subscribe my channel. And I will give you a brief about the PCR in my next video. So till then, have a very good day. Stay hygiene, stay healthy, and keep promoting my channel. Thank you.